Good afternoon, everyone. I'm Catherine Argyriou, postdoctoral fellow in Dr. Nancy Braverman's lab at McGill University and longtime peroxisome disease researcher.
And I'm Corin Chapman. I currently serve on the board of directors for the Global Foundation for Peroxisomal Disorders. When my son Max, who's in the pictures here, was 15 months old, he was diagnosed with Zellweger spectrum disorder. At the time, the geneticist told us that he would lose whatever limited vision and hearing and mobility he had until we lost him entirely. He apologetically explained that there was no treatment. All we could basically do was take him home and love him as long as we had him. Thankfully, that day, he also threw us a bit of a lifeline, and he connected us with a family that had received that same diagnosis, that consequently connected us with the Global Foundation for Peroxisomal Disorders, or the GFPD. And I now know, thanks to the tireless and ongoing work of the GFPD, that we have a real opportunity to separate and change this prognosis for this particular diagnosis. For those of you that are familiar with Zellweger spectrum disorder, it's caused by defects in peroxisome formation and function. It is caused by mutations in one of 13 PEX genes. It occurs in 1 in every 50,000 live births, and 70% of them are caused by PEX1 gene mutations. Now, it can have a broad spectrum, but the hallmark symptoms are limited vision and hearing that becomes progressively worse. Hypotonia, neurological issues such as seizures and leukodystrophy, global developmental delays, and general kidney, bones, and liver issues. Now, I did say it is a spectrum of severity, so those maybe with a more mild version of ZSD may just have the progressive vision and hearing issues, while those with a more severe phenotype are usually babies that don't live past the first year of life. I can speak from experience that those that live more than a year usually have severe disabilities and require round-the-clock care. But luckily for families like mine, the GFPD has been mission-focused for the last 11 years to improve the lives of individuals with peroxisomal disorders by funding research, championing scientific collaboration, and empowering families and professionals through educational services and programs. And it's due to these programs and services and initiatives that we've really been able to get some fundamental research done around peroxisomes and their impact on the body, but also start to tackle the multitude of issues that come with peroxisomal dysfunction. Some of those initiatives include the formation of a world-class scientific advisory board led by Dr. Joe Pisia, who studied many years under Dr. Francis Collins at the NIH. We have an ever-expanding patient registry that has over 650 families in over 40 countries. And thanks to the advocacy of the GFPD, peroxisomal disorders are now included on newborn screening in 20 states, and that number continues to grow. So we're finding these children that have this disorder faster and earlier, even if they may be asymptomatic at that time. We've also been funding a natural history study that's looking both retrospectively and prospectively at the manifestation of peroxisomal disorders in organs. We've funded mouse models at Jackson Labs. And then finally, most importantly, what we've come to speak to you about today is we've made significant strides in retinal gene therapy as well as small molecule treatments for peroxisomal disorders. So at this time, I'm actually going to turn it over to Catherine to talk through the science, but also to really talk about the opportunity to those available who may be interested to partner with the GFPD to take this fundamental research and make it into tangible treatments that provide hope for families like mine.
Thanks, Corinne. So as Corinne mentioned, patients with ZSD can be affected by a multi-system manifestations. But not every patient will have multi-system involvement. However, every single PEX1 patient will have progressive hearing loss and retinopathy that leads to blindness. This is a significant disability for ZSD patient, and the current standard of care for vision loss is supportive only. It includes eyeglasses and walking canes. Improved vision would result in greatly improved quality of life for those affected, as it affects communication, learning, mobility, autonomy, and employment. So to address this, we propose 2 treatment approaches for PEX1-mediated ZSD. The first is a retinal gene therapy, and the second is small molecule therapy. Our gene augmentation therapy uses an AAV8 vector to deliver a normal PEX1 gene by subretinal injection. This is a first-in-class treatment with no competing work. The model system we used to develop this therapy is a well-characterized murine PEX1 G844D mouse model that has the murine equivalent to the most common human mutation. This model was developed and phenotyped by our research team. Rescue in the mouse model includes improved peroxisome functions, improved retinal structure, and the clinically relevant endpoints of improved visual function and functional vision. Success for PEX1 retinal gene therapy would support a platform approach for using a similar approach to target other organ systems and also other PEX gene variants, I should say, which would have a common mechanism of disease. Retinal gene therapy is clinically validated, as we saw with the groundbreaking FDA approval of Luxturna and the subsequent acquisition of Spark Therapeutics by Roche. So this proof of concept data allowed us to win venture capital investment by Amerchem Therapeutics based in Montreal. It allowed us to make an IND or a pre-IND data package that was VC backed. This allowed us to engineer and produce new clinically translatable vectors, which we used to complete in vitro studies, extensive in vitro dose response studies in both human and mouse cell lines. In vivo, we expanded our structural, functional, and biochemical endpoints and performed a dose-finding study to determine the optimal dose to use for our expanded long-term preclinical efficacy study that looked at retinal electrophysiological response, functional vision, peroxisome metabolites, and retinal structure in both the neural retina and retinal pigment epithelium over the course of 7 months in the mouse. So with this data package completed, what's next? So we're currently planning IND-enabling studies and clinical translation. That means we're sourcing toxicity and biodistribution studies from CROs, and we've also begun planning what a clinical trial would look like, selecting potential clinical endpoints, and we are soon launching a prospective natural history study for vision. Our current investors are interested in continuing their financial support for the next round. However, we would require an industry partner or additional investment to make this all possible. If gene therapy is not for you, then we also have small molecule therapy. Our lab developed a functional PEX1 cell-based screening assay that is agnostic of drug mechanism of action, so it would pick up any drug that recovers peroxisome functions or peroxisome import. Multiple small molecule libraries were screened at NCATs using this assay, and several hits were identified. One such hit is diosmetin, which we discovered through subsequent validation and structure-activity relationship studies in our lab, and this best recovered peroxisome import. It's a flavonoid approved by the EMEA for clinical use, including in pregnant women. We tested its efficacy on many different PEX mutations and found it's effective in select mutations in PEX1, 6, and 26, which cover the majority of patients. It's become the gold standard positive control for additional drug screening in many different labs. We've recently had it reformulated to improve its bioavailability in the mouse so we can begin in vivo testing. Recently, a company was founded around this asset, but following a subsequent shift in company strategy, this asset does remain available unencumbered. Other hits include naltrexone, which is a delta opioid receptor antagonist. It was also identified by the NCAT screening. However, we also have other therapeutic modalities in development, including siRNA and CRISPR gene editing that we'd be happy to discuss confidentially with you. So in short, we have multiple leads available to partner for drug development. To reiterate some of what Corin had said, The GFPD and our team have access to a large patient population, the majority of which is actively engaged in our natural history studies. And now, with newborn screening, we'll be able to pick up patients even at the asymptomatic stage. So to conclude, we have all the pieces in place to treat ZSD. We know the target and mechanism of disease, we have the solutions, we have the world experts, and we have the patients. So with that, I'd like to thank you for listening and invite you to contact Corin or myself if you are interested in learning more about how to treat peroxisome diseases and the potential investment or partnership opportunities. Thank you.
Thank you so much.